It will be a short, concise video, but we will cover the most important details you need to know about. Now, before we start, I would like to briefly talk about mycobacteria itself. It's called mycobacteria because it contains a unique substance in its cell wall called mycolic acid. Mycolic acid contains lipids, making the cell wall of the bacteria a lipid-rich cell wall. Mycobacteria are also called acid-fast. This is because they are resistant to decolorization by acid after staining with carbofucsin. Therefore, they gram stain poorly. So, what do we use to stain them then? We use something called Ziehl-Neelsen stain. This stain is used to detect mycobacteria, especially TB. Out of all Mycobacterium species, namely Mycobacterium avium complex, Kansasii, lepri, and tuberculosis, TB is the most important one and the one we will be discussing today. The name tuberculosis comes from the word tubercle, which means round nodule. Therefore, tuberculosis means multiple round nodules. Now let's talk about the features of TB. First of all, it's an obligate aerobic bacillus, which means it requires oxygen to grow. And that is why it makes sense for TB to prefer going to the lungs. Primary infection usually occurs in the lower or middle lobes of the lungs, while reactivated TB prefers the upper lobes, as it has a high VQ ratio. V stands for ventilation and Q stands for perfusion. This means there's a lot of extra O2 and ventilation present, and that is perfect for bugs like TB. Secondly, it's a facultative intracellular organism. This means they can live inside other cells when they need to. And one thing you need to always remember is that TB infects macrophages.